Hi, everybody. It's a real pleasure to be here today to talk to you about fecal microbiota transplants and some of the work that OpenBiome and Finch have been doing. I'm Kevin Roelofs. I'm a research scientist at Finch working on microbial isolation, and I'm really excited to tell you about our shared mission to harness the power of the human microbiome. We'll just start off with a few basic facts about the human microbiome. So this is one of my favorite artistic depictions of the human microbiome. It just kind of shows off the huge amount of bacteria that are covering every surface of us. And they're on your skin, they're on your teeth, they're all inside of your alimentary tract. And basically, although they cover every surface, the majority of them are in the intestine. So today, I'm going to focus on the gut microbiome, because that's the major site of the human microbiome. So what does this tract look like? We have about 7 meters of small intestine, 2 meters of large intestine, and about 32 meters squared of mucosa surface for the bacterial to adhere to along this tract. Within the large intestine at the colon, the bacteria reach densities of 1E11 to 1E12 cells per milliliter. This is one of the most dense microbial ecosystems on the planet, and it's right inside of you. And in total, we think that there's about a little over about 100 trillion bacteria per person that are residing on you and making up your microbiome. But the bacteria are not just the cells, they're the genetic content they encode as well. And here at Google, I'm particularly delighted to talk about the microbiome, which was originally coined as a word to describe the genetic content of a community of organisms. Because actually, we started learning about these biomes through sequencing their DNA. And it's really the information content that I think is at the forefront of understanding what the microbiome is doing. So in total, we've cataloged about 10 million genes that are present in gut bacteria. In any given person, you have about 4 million. And if you remember back from your high school biology textbooks, humans have around 20,000 genes. So the bacteria genetically outpace us by almost 200-fold. As we started to learn over the last decade, really, the research has picked up along with the advent of next-generation sequencing technology. The microbiome has incredible effects on metabolism, immunity, and cognition. And I just kind of label these three as areas where the research is pushing out the farthest. But I would challenge you to talk about areas of human physiology and well-being that don't touch on metabolism, immunity, and cognition. One fact I would like to set straight for the record is you might have heard originally that there were 10 times more bacterial cells than human cells. It's one of the favorite things that we started talking about in the microbiome space. But unfortunately, this is not quite true. And some great work in 2016 estimated all the cells that are present in a human and all the cells that might be present of bacteria, and found that the ratio is much closer to 1 to 1. And actually, one of my favorite lines from this paper was that with each defecation event, you may flip the ratio to favor human cells over bacteria. So every poop matters. And really, one of the things I really like to think about to help visualize the diversity is take a look at this updated tree of life from 2016. So this was a great paper that came out that kind of shows off the major kingdoms, including the bacteria in the top are in blue, are red archaea, and eukaryotes in green. So in that tiny tip that I've drawn a red box around, we have our animal, plants, and fungi. So that's an incredible, just think about it, that's an incredible diversity already. And the distances on this tree reflect evolutionary distance. So if the animals, plants, and fungi are very diverse, but they only occupy that tiny little space, imagine how different all the rest of this microscopic life is from each other. It's a really hard concept to imagine because they all look like single cells, but they're incredibly different in their membrane structure, their enzymes, their physiology. And so this is showing all bacteria, or all life that we know about. If you check out the right side of that tree, you'll see all those red dots indicating that we've never isolated or cultured a microorganism from major families there. But I pointed out a few members of the human gut microbiome. So in the bottom left, we have our E. coli, so the gamma proteobacteria. These guys are one of the first model organisms for microbiology, easily cultured from the human gut. Coming up, we have the verrucomicrobia, which contains members such as Akkermansia mucinophilia. Mucinophilia for mucin lover. This guy hangs out on the mucosa and is a mucin specialist. Above there, we have the most numerically dominant members of the gut microbiome, the Bacteroidales, specifically the genus Bacteroides. These guys are less diverse, but the most abundant members of your human gut microbiome and polysaccharide utilizers. Continuing on over to the right, we end up into the gram-positives, so it's the actinobacteria. And there you have examples such as bifidobacterium, which are some of the first colonizers of the infant gut. And further to the right, the Firmicutes, a very diverse member, or family of bacteria which make up the most diversity of the human gut, although they're less abundant than the Bacteroides. I should also mention the bacteria are not the end of the gut microbiome. You also have eukaryotes present, as well as archaea. And particularly, the archaeal methanogens are very important for nutrient cycling in the gut. But today, we're going to focus on the microbiome as a whole entity. And what I think it's one of the most applicable analogies to it is that it's a microbial organ system. And so you start off, unborn fetuses are sterile, and at birth, they're colonized, either from the vaginal canal or from the skin during cesarean section. And so you can see that what these little circles are that are associated with each of these life stages are compositions, showing off the amount of Firmicutes in pink, the Bacteroidetes in light blue, the actinobacteria in blue, and proteobacteria in red. And the microbial organ system undergoes development as you develop. So as a baby, it's a nascent organ that's mostly dominated by bifidobacterium. With the introduction of solid food, the Bacteroidetes start showing up. As your time in adolescence, you may be subject to antibiotic treatment, which will alter the composition. You may also be malnourished, which can delay the development of your gut microbial organ. Furthermore, by the time you transition through puberty and into adulthood, your microbiome stabilizes and becomes uniquely you. Each of you has a distinct microbiome that's almost as identifying or more identifying than a fingerprint. And some of the major differences we can see in the composition in adult have to do with obese versus healthy, some macro differences. And then finally, as you become elderly, the diversity of your microbiome starts to decline, and we're not quite sure what the effects of that are, but people are looking into it, and it may have, we may be able to help ameliorate some of the effects of old age through restoring microbiome. So it's a microbial organ, and similar to your other organs, it's structured both by your environment and your inheritance. So here I've just shown off, I've talked about a few of the features that impact the structure from the environment, but others, particularly at the infant, are cesarean versus vaginal, antibiotic exposure, breast milk versus formula feeding. There's particular milk oligosaccharides in mother's milk, which only bacteria can digest. And some of the milk formula providers are starting to try to put these sugars into their milk formulas now, but they've been missing in the past. There's also additional factors such as BMI or weight gain during pregnancy. And the results of all these, and I mentioned also you get initially colonized from your mother. The result of this inheritance and environmental factors produce distinct gut microbiome compositions in different populations. So shown on the right is a principal component analysis showing different gut microbiome compositions from United States adults, Malawian adults, or Amerindians. And here you can see that the US population is distinct from the Malawian and Amerindian. Despite having everyone having a unique gut composition, we have a lot of functional redundancy. So shown on the left is the composition by phyla, with the Bacteroidetes in blue, and the Firmicutes in green, and Proteobacteria in yellow. And these are just adults from the Human Gut Microbiome project. And you can see that there's like people who are dominated by Bacteroidetes. There's some that have a balance of Firmicutes and Bacteroidetes. But despite these differences in composition, all of them have almost the exact same functional genes present in their gut microbiome. So when I think about this, I think about it as maybe two different junkyards, one of tractors and one of cars, but both have the functional wheels and engines, is an analogy I like to use here. One of the things that really got me into gut microbiome research was even through my graduate, for over 100 years since snow and cholera, bacteria have been seen as a problem, a disease-causing vectors that are just harmful to humans. And that's really catalyzed a lot of research into bacteria. But it leaves people with the impression that bacteria are bad. And one of the great things about the microbiome is it demonstrates that not all bacteria are bad. Many of them are commensals. They just live on us, and they don't really do much. They don't interact with us much. And some of them are even beneficial. And we establish these symbiotic interactions between beneficial bacteria and ourselves. So we have our cast of characters like Sal Gotcha Manila. That's when you've eaten the potato salad that sat out too long, and others. And so what's the secret? Why can we coexist with all these bacteria, many of which could be harmful to us? And I think the answer comes from from a poem by Robert Frost, that good fences make good neighbors. And so here you see two neighbors making a wall together. And they talk about, and the poem talks about how building this wall helps encourage friendly behaviors, even though it's setting up a barrier between them. And so similarly, there's a wall in the gut microbiome. Shown here is one of the most fascinating images I think have been produced of the human gut microbiome. Actually, this is a mouse model of the gut microbiome, but with a complex flora. The big green section is the mucin barrier that kind of serves as a no man's land between bacteria and eukaryotes. In the blue is DAPI staining of the eukaryotic cells, their nuclei. And in the top left, you see this huge mixture of bacteria, all different kinds mixed together. And you can see that the mucin is almost completely devoid of bacteria. And it's even looking like a laminar structure. And you can see that one of the laminar pieces is sloughing off and starting to be digested by the bacteria. So the host makes the mucin, and it serves as a barrier to protect the host from constantly interacting with microbial signals which could be harmful. I think I'll just say a little bit more about this mucin barrier, because it's just really incredible. It's not only a physical barrier by just being too dense to enable penetration, it is a chemical barrier in that antimicrobial proteins and other antimicrobial particles are secreted from the host into the mucin and serve as a chemical deterrent to microbial colonization. We've now been able to show that phages, which are viruses that eat bacteria, preferentially adhere to mucus and serve as little like, I don't know what you'd call them, bombs in the mucus, waiting for any bacteria to come too close to them. So you have chemical, you've got phage barrier, and in addition to these antimicrobial properties, it also serves as a major source of nutrients for the bacteria. And so that mucin that's sloughing off is actually being digested and promoting, the mucin is decorated with particular sugars that promote the metabolism of beneficial bacteria. So this is just really an incredible barrier. But I think it also helps to show you that there's an incredible milieu of bacteria that are functioning, and they're separated from the host by this mucin. One more picture from the same paper. I mentioned that the mucin can serve as a food source. And so shown here on the left are mice that are fed a standard diet that includes polysaccharides and fiber. And then on the right is mice that are fed a diet that's deficient in fiber. And you can see that the mucin barrier in the right is dissolving. The microbes, without their dietary fiber, are starting to eat away and penetrate further and further into the mucin. So eat your fruits and vegetables, folks, for your gut microbiome. So what particular aspects do we know about the human gut microbiome and health? I think in 2006, it started really kicking off with Peter Turnbaugh from Jeffrey Gordon's lab, showing that you could take obese stool from humans, put it into a germ-free mouse, and the mouse would develop obesity. You could take stool from a lean human, colonize a mouse with it, and then that mouse would remain lean, suggesting that there was obesity-associated gut microbiome. And we went on to find more things about the gut microbiome. 2008, Sarkis Mazmanian showed that there was a microbial symbiosis factor, a polysaccharide that was produced by one of those beneficial bacteria that was highly anti-inflammatory and would prevent autoinflammatory diseases such as inflammatory bowel disease. We found that some bacteria can outcompete pathogens, such as bifidobacteria. We find that, as I said, the gut microbiome develops, and if you're malnourished, it doesn't develop. And in fact, when you go to malnourished children in Malawi, and you try to give them ready-to-eat foods like peanut butter, they don't get better. You need to repair their microbiome before the food will allow them to become not malnourished. There's also, more recently, we've known that microbiome is converting chemicals that are derived from red meat into, the microbes are metabolizing them into a chemical called TMAO that promotes atherosclerosis, otherwise known as heart disease. And finally, there's some great work going on also in the Mazmanian lab by Elaine Zhao, demonstrating that some of the microbiota differences you can observe in mice lead to neurodevelopmental disorders. And so this is an autism spectrum model in mice. And additionally, I think we're also finding type 1 diabetes, another inflammatory autoimmune disorder. So you can see that this is having to deal with metabolism, immunity, cognition. One of the diseases we know about the most in the microbiome is Clostridium difficile. C. difficile is a hospital-acquired infection that we heard about. It can kill about 30,000 Americans a year, costs $5 billion in hospital costs, and it's been rising. Although I think actually, due to OpenBiome's work, it's been falling lately. With C. difficile, you can take antibiotics, but it's a spore-forming organism. So even if you are able to remove the vegetative cells, the spores are antibiotic-resistant, and they can remain in the gut and then germinate again. And so antibiotics have some efficacy, but they don't cure. So about 30% of the time, they'll be effective in curing C. difficile. And the way this is working, I think I'll just explain this little diagram here. I think this is a really appropriate model for fecal microbiota transplant. You start with a healthy microbiome, which can be perturbed by antibiotics, which we need to take because some of these bacteria are pathogens and they'll kill you. So you need antibiotics. Let's not make any mistakes about that. But as you're doing that, antibiotics are like nuclear bombshells on the microbial world. They're just killing everything. And hopefully, and most times, the bacteria are able to grow back and restore a healthy microbiome. But sometimes with this clear cutting of the microbial ecosystem, you can get a weed like C. difficile infecting and overgrowing and preventing return to a normal healthy microbiome. And this can lead to more antibiotic treatment to get rid of the infection, which is causing horrible diarrhea. But then that leads right back into another round of C. difficile disease. And so one of the most effective treatments you can do is take a whole fecal microbiota transplant, put it into a person, and just restore the healthy ecosystem. And that actually has about around a 90% cure rate for C. diff. It's an incredible improvement over antibiotics. But one of the problems is access, because as you mentioned, FMT used to be pretty sketchy. People would get blenders and give each other transplants by having their roommate poop into a blender, grind it up, and then give themselves an enema. That's not a very safe way to do fecal transplants. But I'm showing here a picture of Mark Smith and James Burgess, the co-founders of OpenBiome. Mark had a friend who was suffering from C. diff, and he just did not have access to a clinician who could provide a fecal transplant. The clinicians are worried about whether it's safe. They don't know how to do it. But Mark was working on this in the lab, and he knew that FMTs could help his friend, but he just couldn't get them from anywhere. So with James, they decided to make OpenBiome to provide access to FMT using a universal donor model. So the universal donor model, what is that? That's the model where instead of trying to find one specific person who's maybe you live with so that you'd have a similar gut microbiome, and trying to make sure that they don't have any infectious diseases that you could get, and screening that one person individually, and then trying to get a fecal transplant done. Instead, what you do is you open up a stool bank, and you heavily screen donors, and you enable everyone to provide, enable very healthy donors to provide stool that can then be given to clinicians to just be used as a treatment, instead of relying on the clinicians knowing about how to do the screening and how to do the preparation. And so OpenBiome has been incredibly successful at scaling the universal donor model. They've provided over 25,000 treatments in 950 hospitals and clinics in OpenBiome's network, in all 50 states and in 7 countries, including the US. And as part of this, the Universal Stool Donor Program, that's kind of why I'm here today, to help tell you about this and encourage you to become a part of this program. There's an incredible drop-off because there's a lot of screening that's done. There's a pre-screen survey in which about 66% of people pass, just looking at lifestyle-associated conditions. There's a clinical assessment in which 84% make it through. And then finally, laboratory testing, which results in another only 47% getting through, to result in a total of only 2.8% of our donors who apply being able to be a stool donor. So this is a very tight pipeline, but it can be very effective. And so I'd encourage you to give poop at givepoop.org. You can save lives, earn money, and donate your stool, and make your morning routine heroic. I think one of the most exciting things that we're doing at Finch and OpenBiome is looking ahead to find out what new diseases might fecal transplants be effective for. And so I'll just share with you a small vignette about alopecia. Alopecia is an autoimmune disease in which you start to lose your hair, or all hair on your body. And so shown here is a patient at 16 years old on the far left, starting to lose hair. Then this patient, unfortunately, also contracted C. difficile, and then received a fecal transplant as a result. And so this middle picture shows him right before receiving the fecal transplant. And then after the fecal transplant, he noticed his hair started to regrow. And this one anecdote has now become 2 anecdotes, and we're very interested in following up on it, as for one example. But this kind of shows you how connected the gut microbiome is to our overall health in ways that we can't know today, but we're hoping to discover tomorrow. And so with that, I'll leave you by saying that OpenBiome and Finch are focused on safe access, clinical research, perpetual access, and new cures. Thank you. Now I have some time for questions and answers. And I'd like to also introduce Meg Muckenhoupt. Did I mispronounce your last name horribly? Yeah. But Meg is on our external affairs team at OpenBiome. Yeah.
We screen for a lot of different conditions, and you do have to have a BMI under 30 if you want to become a stool donor. In the front.
So why is the pipeline so narrow? Is that you really have to be that careful? Is that an abundance of caution? Like, what makes it so narrow?
Why is the pipeline so narrow? Well, partly because there's research indicating that the gut microbiome is related to pretty much everything. Partly because a lot of people tend to carry conditions they don't know about. When we test for stool donor, the people that we reject, that don't make it through, rather, aren't necessarily unhealthy, but they're people who are carrying a transmissible disease or some sort of condition that we think might harm the people with C. diff. I mean, 2 things to remember here. 1, we're regulated by the FDA, which has very, very strict standards. And 2, the people who are getting fecal microbiota transplants are really, really sick. You know, fecal microbiota transplants are only approved for recurrent CDI, C. diff infections, not the initial C. diff infections. So by the time they get to us, they've already been through at least 2 rounds of antibiotics, typically had this disabling diarrhea for 3, 4, 5, 6 months, or even longer in some cases. So we can't take any chances that, oh, that little norovirus that you happen to carry that doesn't cause you any problems could kill this person who's already in a very delicate state of health. So that's why it's narrow, and we can't predict who's going to make it through the pipeline or not. You know, some people are gung-ho rowers and eat a perfect fiber diet. Some people just kind of munch French fries and walk around, but they, for some reason, have this really great gut microbiome and aren't carrying anything we don't want in there. We can't predict. That's why we have to ask all of you to apply. In there?
Do you have to match donor and recipient, like blood types or whatever?
Do we have to match donor and recipient? No. That's the wonderful thing. As long as the person isn't carrying some sort of disease or has some sort of condition we think might make you sicker, pretty much anyone will do. It's really amazing. It'll all work on C. diff, and it works about 90% of the time. That is not necessarily true for other conditions. OpenBiome, we have a dual mission of providing safe access to fecal microbiota transplants for C. diff, but also promoting clinical research. And we're currently involved in 30 different clinical research trials looking to things like inflammatory bowel disease, Crohn's disease, even peanut allergies in one case. And there's some indication from some of our work on types of inflammatory bowel disease that there are differences between donors and their effectiveness in promoting remission. So for C. diff, it doesn't matter. For other things, it might, but we're not sure yet, which is why we're doing all this research.
Hi, wonderful talk. I was wondering if you could tell us anything about what's either known or theorized about the mechanisms between the gut microbiome and any of these other conditions, such as obesity or the hair loss, or what's either known or theorized about those connections.
So sometimes we're digging into specific mechanisms, and sometimes it's not known. So particularly in the obesity case, there was a investigation into the metabolic genes, and we were able to see that there is an overabundance of metabolic genes associated with energy harvest, suggesting that potentially the microbes are just liberating or making, so you have this fiber that normally is just excreted and used as food for the bacteria, but they may be producing metabolites which can then be absorbed by the human host, and that may be providing more energy from a given set of food. And actually, those metabolites are also potentially underlying some of the inflammation parts. So I mentioned there was a specific polysaccharide that was released by Bacteroides, which was anti-inflammatory. But that's a pretty specific one. One of the most general mechanisms that's well known is the production of these short-chain fatty acids, like butyrate and propionate, which are just small little carbons that result from the breakdown of polysaccharides. And many bacteria in the gut can release those, and they serve as actually the primary food source for your colonic endothelial cells. And so not only do they help the cells by providing their preferred metabolite, they can also trigger specific receptors, like G-coupled protein receptors in immune cells, to have diverse effects, but the results of which are generally the production of anti-inflammatory cytokines and the reprogramming of immune cells away from inflammation towards tolerance.
And people are looking at these immune effects of the gut microbiome in an astonishing number of conditions. There's some really interesting research coming out about the gut microbiome and multiple sclerosis and Parkinson's disease, and there are a couple of really interesting papers that came out just last week about mediating in cancer treatments that involve the immune system, that some people who have certain type of gut microbiota, chemotherapy is much more effective. And if you take their gut microbiome and put them in germ-free mice, you find the same differences in the mice as you did between people when they undergo cancer treatment. So anything having to do with immune disease, just anything, it's pretty astonishing.
So there was a paper in Nature, I believe 2014, about the effect of sugar substitutes on the gut microbes and resulting metabolic syndrome symptoms, essentially, which could be transferred from a mouse to a mouse by fecal transplant. Seems like the sort of thing that we both known to you and flashy and shocking. We've been followed up by other researchers by now. What's the latest in that area?
So I actually know that paper pretty well. It was fascinating because it's like what are these are artificial sweeteners. They're supposed to be not metabolized, right? So they're supposed to be calorie-free, but still have sweetness. But one of the that means by not metabolized, normally sugars are like digested, and the simple ones are dissolved almost at your mouth, like, and if not by then, at least in the small intestine. But these ones are not, and they make their way straight down into the gut microbiome, the colon. And there, they were shown to have an effect on promoting signaling that would lead to like metabolic disorder in mice. I haven't seen that there's been new updates on that story. I think one of the caveats there is that I tried to calculate how much artificial sweetener they were feeding the mice. And I think it was somewhere around, like, if you were a human, you need to have like 70 packages of, God, which one was it? I won't say which one so I don't get in trouble, but like whichever one it was, you'd need to open 70 packets and eat that much to get the same dosing that was being applied to the mouse. Also, the other interesting part is they didn't just feed it to the mice. They took the bacteria out and they dosed the bacteria. They grew it with this artificial sweetener as the sole nutrient source for a while. And then they transplanted it back, and then the community had this different signaling effect. So there's a few caveats there, but it definitely hinted that these artificial sweeteners may not, while they may be calorically inert, they may have a strong signaling effect mediated by the microbiome.
Fun fact about mouse experiments in the microbiome. Mice do their own fecal transplants if they're housed together, much like dogs do sometimes. There was another question in front.
I was just wondering if you're doing any research on irritable bowel syndrome.
Research on irritable bowel syndrome. We certainly are. If you're interested in any clinical trials of fecal microbiota transplants, clinicaltrials.gov. You can put in on the search term your condition and then FMT or fecal microbiota transplant or just fecal, and you'll come up with a list of trials. I know there are a couple running in Boston, probably Dr. Jessica Allegretti at Brigham and Women's, but don't quote me on that. Look it up on clinicaltrials.gov. Any other questions?
Could you speak more as to what you're looking for in the pipeline that you mentioned earlier?
So at each of these stages, I think I mean, Maggie, you might be the right person to answer this. Yeah. Like,
on a regular basis?
The donor survey has a variety of questions about lifestyle risk. So maybe, are you monogamous? Do you try?
No?
Oh, okay.
Honestly,
They've updated it.
yeah. We really want to know that you live in Boston or Cambridge or somewhere around here, first of all, because we get a lot of people applying who say, this is cool, I live in Paraguay. Like, no, you cannot send us your poop in the mail. Don't even think about it. So that's our primary criterion. Can you actually get to one of our donor sites? And we have donor sites at Innerbelt Road in Somerville near Sullivan Square Station, and one in Central Square, very convenient to Google headquarters, and another one at BU West if you're on that side of the river. And they're open every day of the week, and they're very convenient, and they're staffed by friendly people. You know, we're looking to see you're healthy. If you have a history of colitis or inflammatory bowel disease, this isn't what you should be doing. But overall, it's about health, and again, there are things that you may be carrying. A lot of people carry parasites without knowing it, you know, hominids or something like that, and you can't tell that until you've gone to the end of the clinical interview and gone through blood and stool screening, because you wouldn't ask your doctor. You know, he wouldn't say, hey, can you just test me for parasites? You have no reason to suspect, but again, you could be carrying something that could make someone very sick. We test for more than 30 different conditions, and we have a rather lengthy questionnaire that people go through before they even go to the nurse. So it's hard for me to summarize it right now, but if you're curious, go to givepoop.org, register, and we will probably point you to a copy of the questionnaire, and you can see for yourself. Other questions?
That just prompted one. When you say you do all the testing, do you give the results to the donor, or is that just used internally?
If we abide by HIPAA regulations, and if we find anything new about your health that you do not tell us about, like if we get the stool and blood sample back and say, hey, wait a minute, this person is positive for HIV, for example, we certainly tell you. But if it just comes back that it's something you said, oh, by the way, I got hepatitis last year, and you come back positive for hepatitis, we would call you. But generally, we don't find anything new. Most people are excluded from donation, not because of anything new, but because of something that could affect the people who are getting the transplant, like having a family history of colitis. Yes.
I came in late and missed the talk, so I'm going to ask a question which might have been addressed. What assays do you perform on the samples? Genetic assays, sequencing, such?
Yeah, that's a great question. So, as I mentioned, the reason we start to know so much about the gut microbiome is because of next-generation sequencing technology. And so, particularly the two types of sequences we generally perform are 16S amplicon sequencing. So here, every bacteria has a specific gene that's called the 16S ribosomal RNA gene. And here, because each one of them has it, you can use it as a marker for how they're related to each other. And that can give you an overall picture of the community. But it doesn't get a high enough resolution. Because everyone has it, it doesn't change fast enough to really let you get down into the species, much less the strains. So bacteria with identical 16S gene sequences could have up to like 20 to 30% of their genome be different. So the other type of sequencing we do is called shotgun metagenomics. And here, instead of starting with a PCR, a polymerase chain reaction to target a specific gene, you just take every single piece of DNA that you can find and start sequencing it. And that gives you a piece here, a piece there, and they're hard. It's much more computationally difficult to understand the results of shotgun sequencing, but it can get you down into the strain level composition of the gut microbiome.
I'll follow up on that and ask exactly what do you do? I mean, what assembly technology do you use to try and assay these shotgun sequences? I used to work at the Broad Institute across the street as a building genome assembly tool, so that's my interest.
Yeah, unfortunately, I don't think I can say much about that for two reasons. One, I'm mostly on the microbial isolation side. I'm really hoping to improve my computational skills, but I just couldn't speak confidently about the different pipelines that are being used right now by our data science team. I'm happy to be put in touch with Sonia Timberlake, who is our director of data science, who could maybe say some more about that.
Thank you very much.
Thank you.